You are welcome to our concluding biology class on photosynthesis. For this class, we'll be looking at experiments to test for factors involved in photosynthesis. Now, in our previous class on photosynthesis, we have learned that during photosynthesis, carbon dioxide combines with water in the presence of sunlight energy and chlorophyll to give rise to glucose, while oxygen and water vapor is given off. Now, we would like to note that the carbohydrate or glucose that is produced is stored as starch in the plant. Therefore, the evidence to show that photosynthesis has taken place is by detecting the presence of starch. When the glucose formed is stored in the plant, it is stored in the form of starch. So the only evidence to show that, or the ultimate evidence to show that photosynthesis has taken place, is by detecting the presence of starch in the leaf of the plant. Now, starch grains appear this way when they are stained in being embryo. Starch grains of starch, when it is busted and stained in being embryo, this is the way it looks like. Now, before we describe this experiment, there is pre-experimental knowledge that we must have. One, the test and control experiments use detached plants. The test and control experiments use discharged plants. Plants are discharged by keeping them in the dark for 2 days. That is, discharging is that you remove the starch that is already stored in the plants before you remove the plants from the field. So if you take a live plant that is on the field and you use it directly, you won't be able to confirm that it's actually the photosynthesis that we have monitored that is responsible for the starch in the plant. So what we do initially is to discharge. We call it discharging. We discharge the plants by keeping them in the dark for 2 days. Then plants are tested for the presence of starch at the end of the experiment. So it is absolute that no starch will be present when a starch has been put in the dark, when a plant has been put in the dark for 2 days, for 48 hours. The starch stored in the plant will have been used, during the respiration that takes on or that takes place when the plant is kept in the dark. Now, to test for starch in a leaf, 1, remove a green leaf from the plant. Boil the leaf in boiling water to stop enzymatic reactions within cells in leaf. Because sometimes in examination you are asked, why is the leaf boiled in water? When you remove the leaf from the plant to test for starch, the first thing you to do is boil the leaf in boiling water. And you do that to stop enzymatic reactions within cells that are in the leaf. Number 3, put the boiled leaf in boiling tube containing alcohol or ethanol. This is meant to extract chlorophyll as ethanol dissolves lipids in cell membranes. So you can see in the picture or experiment shown there, you can see the hot water in the beaker. You see the alcohol inside the boiling tube. Hot water is in the beaker. Alcohol or ethanol is inside the boiling tube. Then we have the wire gauze and the tripod. Then you put a Bunsen burner under it, and it will boil it off. When you boil in ethanol, it extracts chlorophyll as it extracts the chlorophyll in the leaf. Now, 4, that will enable your test to show forth at the end of the day. Now, 4, return the leaf to the hot water, softens the leaf and allows penetration of iodine. So when you complete the boiling in step 3, you return the leaf to the hot water to soften the leaf and allow penetration of iodine. In examination, you are often asked 3 reasons why in a test for starch in a green leaf, why do you boil the leaf in boiling water? Why do you boil the leaf in alcohol or ethanol? Why do you, return the leaf in hot water after, boiling it in alcohol? The reasons are underlined here. They are very important points for you to note. Then after now, you now remove the leaf and perform the iodine test. Now, when you perform the iodine test, you put few drops of you place the leaf on a white tile and introduce few drops of iodine solution onto it. You are expected to observe either purple or blue-black coloration to show the presence of starch in the leaf. Now, to continue our experiment, we want to look at some hypotheses. If carbon dioxide combines with water in the presence of sunlight, energy, and chlorophyll, glucose is formed, oxygen and water liberated, and the deformed carbohydrate is stored as starch in the plant. Now, if we were to remove carbon dioxide, would there be starch produced? That is the first hypothesis. If this carbon dioxide, which is one of the raw materials, if it is removed, will starch still be produced? Now the second question is, what if we remove chlorophyll? Will starch still be formed? Now, after this time, we are going to test the hypothesis. We are going to test our hypothesis in the second part of our class on experiments concerning photosynthesis.